So in the last video, we discussed what are the different types of DNA repair mechanisms, and the first of them was mismatch repair. So let's see how this mismatch repair is done. In the present video, we'll discuss the mismatch repair in the case of prokaryotes, that is the organisms which don't have a proper nucleus. So first of all, let's see what is the structure of our DNA. See, DNA we have seen, it's made up of a double helix. If the one strand goes from 5' to 3' direction, then the other strand must go from 3' to 5' direction. So in the DNA structure, we all know that the adenine always makes hydrogen bonds with thymine, and the cytosine always makes 3 hydrogen bonds with guanine. So this is how these should be paired in the case of correct DNA structure. Now suppose there is some mismatch, like in the present example, suppose this is the correct strand and here we have seen it's guanine there, then it had to be paired with cytosine. But in the present example, this has paired with thymine. So there is some sort of mismatch here. Due to this mismatch, there is some distortion here. That's why this strand has become like this. It has become distorted. So we have seen there is some mismatch, but we don't know whether thymine is the correct base and guanine is mismatched, or guanine is the correct base or thymine is mismatched. In the case of DNA structure, there is so many GATC segments, that is segment of the 4 bases, where first one is guanine, then adenine, then thymine and then cytosine. These are called GATC segments. There are so many and so many GATC segments in one DNA strand. They say one GATC segments occur for every 243 bases, or one GATC segment occurs after every 256 bases. So that means in a nutshell, there will be hundreds and thousands of GATC segments in one strand of DNA, and maybe more than that. These GATC segments play an important part in recognizing which strand is the parent strand and which is the mismatched strand or the daughter strand. You can say, or the complementary strand. So how this is done? These GATC segments have got one adenine base, and this adenine gets methylated with the help of enzyme DNA adenine methyl transferase in the case of prokaryotes. In short, we can write it as DAM. What is DAM? DAM is DNA adenine methyl transferase. This enzyme actually transfers the methyl group to the adenine in GATC segments. In the complementary strand also, there will be many GATC segments. As we always read the DNA strand from 5' to 3' direction, so it should be like this. GATC is from 5' to 3', GATC. And here also, there will be complementary GATC. So from 5' to 3', there will be GATC. C will be complementary to G, T will be complementary to A will be complementary to T, and G will be complementary to C. So this is how actually it is. So what the cell does, it sends a protein known as mut-S. This mut-S protein occurs in the form of dimers. So there are 2 mut-S proteins attached to these distorted segments. So mute S protein comes and gets attached to this distorted segment. After that, mute L protein comes and binds over this combination, that is combination of the distorted segment with the mute S2 because it occurs in the form of a dimer. So this is our mute L protein. It comes and binds to this combination of distorted segment with mute S. This whole structure, this combination, it uses ATP and cause the DNA strand to bend upon itself. Now what happens? This GATC segment, it also got bent here. But here there was one protein known as the mute H. Now due to this bending, this mute H protein comes in contact with the mute L protein bound to this distorted portion. So ultimately, now we have got a combination of distorted segment with mute S2, with mute L, with mute H. So this is the structure formed before the mismatch repair takes place. This mute L, it causes the activation of an enzyme known as UVRD helicase. It causes activation of UVRD helicase. This UVRD helicase, it causes the unwinding of this double-stranded portion of DNA up to the GATC segment. Mute H, it takes the methylated strand as the parent strand and the other strand which is non-methylated yet as the complementary strand. Actually, this methylation does not occur immediately after the replication. So, till some time one strand of the DNA is methylated and the complementary strand is not methylated. So now it knows that the whatever base is there on this complementary strand, that base actually is mismatched. So in the present example, mute H is taking this strand from 5' to 3' as the parent strand. So this T is taken as the correct base, and the other strand which is non-methylated, this one where we have got the G base. So now this G base is taken as the mismatched base and we have to remove this base. So let's see how it happens. So when this contact occurs, mute H protein gets activated. Mute H is actually an endonuclease. So when it gets activated, it produces a nick here in the adjacent segment. So it makes a nick here. Now this portion of the DNA has been already unwounded with the help of the UVRD helicase. And now we have made a nick with the help of mute H, which is actually an endonuclease. So after this nick is made, exonuclease enzyme comes here. These exonucleases can be 5' to 3' exonuclease, 5' to 3' exonuclease, which keeps on cutting the nucleotides from 5' end. So in the present example, this exonuclease has to remove the nucleotides like this from the 3' end. Here will be 3' end and here will be 5' end. Suppose we have removed one nucleotide, this is the second nucleotide we have removed, and this is the third nucleotide already there. So it has to ultimately remove the nucleotides from the 3' end. So we have to use 3' to 5' exonuclease. So it keeps on removing the nucleotides from the 3' end till the portion of this mismatched base, and just after that it makes a nick here and keeps on removing the nucleotides one by one, like this. It will remove this one, then this one, then this one, and it keeps on removing the nucleotides just after the mismatched base. So ultimately, now we have got only one DNA strand, that is the parent DNA strand, and this mismatched base and the complementary strand has been removed. So our enzyme DNA polymerase 3 comes now in the case of prokaryotes. As we all know, polymerase always reads the template strand from 3' to 5' direction. That is, it will read the template or the parent strand from 3' to 5' direction like this, and it will keep on adding the nucleotides from 5' to 3' direction. It will read the template strand from 3' to 5' direction, and it will keep on attaching the nucleotides from 5' to 3' direction like this. So it will keep on attaching the nucleotides here, so that a new complementary strand is made with correct base attached here. Now this distortion will get removed and we will have a correct complementary strand. Let's see the whole process again. Suppose there is some mismatch, it causes a distortion in DNA strand. Due to this distortion, protein mut S come in the form of a dimer and gets attached here. Over that, one more protein mut L gets attached, which can cause activation of UVRD helicase. Mut H protein gets attached to the GATC segments present in the DNA molecule. These GATC segments are methylated in the case of parent strand and are not methylated in the case of the complementary strand. So this is how mut H protein recognizes which is the parent strand and which is the complementary strand, because we know whatever base is present in the case of complementary strand, that base must be the wrong one, that base must be the mismatched one. This methylation does not occur immediately after the replication. It takes some time for the enzyme DNA adenine methylase or the DNA adenine methyl transferase to cause the methylation of the adenine present in the GATC segments. So during that period, this mismatch repair is done, because during that period only we can know which is the parent strand and which one is the complementary strand. So now this whole portion, the formation of this whole combination, that is distorted segment plus mut S2 plus mut L, causes the bending of the DNA segment so that the protein mut H comes in contact with this combination. This whole process causes the activation of mut H, and this mut H forms a nick in the complementary strand or in the strand having the mismatched base. So it forms a nick here and acts as a endonuclease. After that, exonuclease comes and keeps on removing the nucleotides from here one by one till just after the mismatched base. After that, polymerase comes and keeps on attaching the correct bases or the nucleotides with the correct base in 5' to 3' direction, so that ultimately we have got a new DNA molecule with no distorted portions. This is how mismatch repair gets done in the case of prokaryotes.